Okay, let's go. So we're going to focus then today looking at human digestion enzymes. I want to look at enzymes. I want to look at the digestive system, because an understanding digestive system when I did all that. And it is highly likely you'll get questions based around the practicals, and the two key practicals for this part of the course are food tests. You need to be able to understand the 4 different food tests, what they are and what they test for. And this one is probably the most likely practical you'll get in the higher paper, which is all about the effect of pH on amylase. So I'll talk you through that as well. Okay, so we've done these questions. The focus for us now is to focus on the digestive system. So starting from the very top, as we all know, my drawings are exceptional. So I'm going to draw a human body. That's meant to be his little nose, which looks like it's broken, which is it's a good start. He's a very odd looking chap. We don't want that to go on our YouTube, Oliver, do we? So now it's going to sound like it's Oliver that I'm drawing, because you're the only name I've said. Okay, so the mouth doesn't really count in terms of a digestive organ. It contains lots of useful things. The key thing to start with is the salivary gland. So the salivary gland we find obviously in the mouth. The salivary gland does produce saliva, but its key function is it produces salivary amylase. We'll talk about the function of all the enzymes next, but if it ends in ASE, it's an enzyme. So any word you see come up that ends in ASE, it is an enzyme. The first one we've come across is salivary amylase. The next one then is this muscular tube. This muscular tube is called your esophagus. And it plays a key role in physical digestion, which means it physically breaks things down from large molecules to small molecules to help them pass through the rest of the digestive system. So your esophagus is a key part of muscular and physical digestion. After that then, the food enters this organ here. And this is my stomach. Okay? The stomach has got lots of functions. Not all of them are related, though, to digestion. And most of you got it wrong when you said what one of the functions was. The first thing, the stomach is a very powerful muscle, which again means it can be used in physical digestion. So it breaks things down from large molecules into small molecules. However, its most important role is to start the process of chemical digestion, where it's beginning to break bonds. So in the chemical digestion, it produces a protease. Again, it ends in ASE, so it must be an enzyme. So it produces the protease enzyme. So it starts to break down proteins into a smaller protein, which we'll look at in a second. And that is a form of chemical digestion. Key thing, and this again is a question they like to talk to you about in the exam, this protease is incredibly well adapted, because the pH in the stomach is between about 1 and 2. No other enzymes will work in the stomach apart from the stomach protease, because it's well adapted to work at that pH. Every other enzyme would be denatured. This one is not denatured. So that goes me on to talk about the next thing, which is hydrochloric acid. Hydrochloric acid is found in my stomach, but it doesn't play a role in digestion, which is a common mistake. It doesn't have any role in terms of breaking anything down. Its pure role is to destroy pathogens. It is a barrier for your immune system. So it just ensures no food you're eating contains a pathogen. That's its function. So it's not a role in terms of digestion. It prevents you getting food poisoning. Once the food leaves the stomach, it enters this very important part here. Okay? This is called the duodenum. And it's a grade 8 or 9 question to talk about the duodenum. The duodenum is the top part of the small intestine. And I'll look at that in a second. The reason it is so important is because of this. At the duodenum, this tube comes in, which I'll talk about in a second. And at the duodenum, this comes in, which I'll talk about in a second. So the duodenum is where 3 parts of your digestive system mix. So it's an incredibly important role in chemical and physical digestion, but particularly in chemical digestion. So let's talk about these bits then that are coming in here. So we have, this is our liver. And the only function of liver in digestion is to produce bile. And I'll talk about the function of bile in a second. Once it's produced it, it stores it in this little pocket here, which is called my gall bladder. And then when your digestive system needs it, it travels through this tube here into your small intestine. I don't think they'll ask you the name of this tube, but it's called your bile duct if they ask you. This little green thing here is your pancreas. It looks a little bit like a bunch of grapes or a leaf, or a combination of a leaf and a bunch of grapes. It is a very, very important gland, and it plays a very important role in digestion. The pancreas produces all digestive enzymes. So if you're ever in doubt, if the question asks you, where is protease produced? Pancreas. Where is amylase produced? Pancreas. Where is lipase produced? Pancreas. Because your pancreas produces all the digestive enzymes. At this point then they all mix together, and then you pass on or continue to work through this thing here, which is my small intestine. Okay? So the food then passes into your small intestine. The small intestine also produces all digestive enzymes. So protease, lipase and amylase are all produced in the small intestine and the pancreas. So they are the main sites of chemical digestion. But it's in the small intestine where all of the soluble particles, and we're going to call them monomers because that means they are small units. It's there where they are absorbed into your bloodstream. What happens next then is the food passes into your large intestine, which effectively goes around the outside of your small intestine. This is your large intestine. Your large intestine is where water is absorbed. And any food that is not absorbed in the small intestine will continue along the large intestine, not to be absorbed, but to eventually produce your feces. And a common misconception here. People think that feces is an example of excretion. It is not. Feces is an example of something called egestion, which just simply means exit. Excretion is a product that is produced. You do not produce feces, it's just products that are not being used. So it's egested, not excreted. Okay, so there are four food tests that we need to know. One of them is for starch. One of them is for sugars, but again, in the higher paper, I would never use the word sugar, I would use the word glucose. Okay, so we'll try and use the word glucose if we can. Then, of course, we're going to have a test for proteins. And we're going to have a test for fats/lipids. So these are the four food tests you need to know, which relate to the 4 different food groups you'd be expected to do. Okay, so starch. What's going on here, guys? Okay. In its simplest way then, I have a test tube. In this test tube, I'm going to add a sample of food. Okay? So I'm going to add this sample of food. In an ideal world, depending on how much detail they ask when they write a method, what you will do, and I'm only going to write it for this one, you use a pestle and mortar to grind up the food. Pestle and mortar is one of those grindy things that grinds them up. Okay. Let's carry on. You talk again, I lose subscribers. Okay. So I have my sample of food, which has been ground up using a pestle and mortar. To make it soluble, because you want it to be soluble, I add then water to it. So effectively I have a little solution of food in the bottom of my test tube. Okay? What I'm then going to do for this experiment to test if there is starch present, I'm going to add iodine. So the test for starch is the iodine test. If starch is present, so if there's starch in my test tube, it will go from a yellow-brown colour, hence why I chose this colour to be yellow-brown, to black. So if starch is present, it will go from yellow-brown to black. Yellow-brown? Yeah, it's like it's a yellowy-brown colour, depending on how strong the iodine is. Weak iodine is very yellow, strong iodine is very brown. Okay? But it's obviously yellowy-brown. The next one then, I'll do proteins next. So proteins, the test is called the Biuret test. So again, I have my sexy test tube. Inside my sexy test tube, I have a solution of food which has been ground up using a pestle and mortar and water. And then I add a few drops of Biuret solution to my test tube. And Biuret solution starts off as blue. If there is protein present, it will go a jazzy, I know what you're thinking, purple colour. P is for purple, P is for protein. So proteins go purple. Okay, so the test for sugar and glucose, we use the Benedict's test. To the test tube where I have my food sample which has been dissolved in water or mixed with water, I add Benedict's solution. You then have to heat it. When you heat it, I would say I heat in the water bath. The test for sugar or glucose is what we call a semi-quantitative test because it gives not only just present or not present, it tells us blue means there's no glucose, green means there's a little, yellow means there's a fair amount, red means there's lots. So it's a semi-quantitative test. The final test then is the test for fats and lipids. And again, I have my food sample which has been ground up using a pestle and mortar and added a little bit of water. I'm then going to actually add more water to this one. Okay? So it now starts to form more of a solution, actually. I then add, after I've added the water, I obviously give it a little shake. I then add ethanol. And again, I give it a little shake. If fat is present at the top of my test tube, I appreciate you can't see that very easily, I form a white emulsion. So at the very top, if lipids or fats are present, I form a white emulsion. And that is why this test is called the emulsion test. Enzymes. An enzyme, every single enzyme, is a protein. So all enzymes are made of protein. All enzymes are what we call biological catalysts. And then from a chemical point of view, or chemistry point of view, a catalyst does two things. It speeds up a chemical reaction, and this is not used up. And enzymes are exactly the same. They are biological catalysts that speed up chemical reactions without being used up themselves. So if I ask you for a definition of an enzyme, I would say it's a protein that is a biological catalyst that speeds up chemical reactions without being used up. If you want to talk more about the chemistry, I don't think they would in a biology exam, but the reason it does that, and I will just make a quick note, is it reduces the activation energy, which is a term you should be aware of in chemistry, which is the energy required to start a reaction. So that is how a catalyst actually works. It reduces the activation energy. Okay? An enzyme also does this. So all enzymes, or digestive enzymes I should say, break down polymers to monomers. So what I mean is this. They break down a large molecule, which is insoluble, to lots of small molecules which are soluble. Okay? Right, so let's talk about the three examples. We're going to set up a little table. So, the table's going to say enzyme, where it is produced, and its function. So the first enzyme is amylase. Again, it ends in ASE. Okay, so amylase ends in ASE. They might, if they're feeling fruity in the exam, not call it amylase. They might call it carbohydrase. They're exactly the same thing. Amylase is just an example of a carbohydrate, which should make our function actually easier to remember if we know it's a carbohydrate, because because then it will link very clearly to our function. Amylase is produced in 3 places. As I've already said, all enzymes are produced in my small intestine, so it's produced in my small intestine. All digestive enzymes are produced in my pancreas, so it's produced in my pancreas. And this one, as we said earlier, is also produced in your salivary gland. So the function of amylase is to break down, or a better word would be hydrolyze, but in GCSE you'd certainly find just saying break down, carbohydrates into sugars. That is a very basic definition of what amylase does. We're all doing the higher paper, so I'm going to give you a slightly better definition, which is it breaks down starch into glucose. And glucose is then used in respiration. The second digestive enzyme is protease, which again is produced in my small intestine, because all digestive enzymes are produced in my small intestine. It's produced in the pancreas, because all digestive enzymes are produced in the pancreas. And this one, as we said earlier, is also produced in my stomach. The function of a protease, very simply, is to break down protein, which is a polymer, into amino acids, which are monomers. Amino acids then are used to make every protein in your body. So we break a protein down to build other proteins, and we need that then for growth and repair. The final digestive enzyme then is lipase, which is produced in my small intestine and produced in my pancreas, because all digestive enzymes are produced in the small intestine and the pancreas. The function of lipase is to break down lipids, and if you remember the word liposuction quite literally means sucking out fat. So lipids is just the scientific word for fats, and it breaks down lipids, and this is the one most people tend to forget, into things called fatty acids and glycerol.